Hello College Biology. Here is our fifth topic in cellular transport, exocytosis. Exocytosis is a type of bulk transport, so we're moving large quantities of particles, and we're going to be using ATP to shape the membrane a little bit. And it is used mostly to expel waste products, but we can also use it to create or to expel cell products out of the cell. All of the sweet things in biology are made by cells, such as spider silk and milk and mucus and the sweet, sweet pigments that make animals cool looking, and certain defenses like venom. All of these are cell products that have to leave the cell if they're going to get used by the organism, in addition to waste products. So how do we get all this stuff outside of a cell? Well, this story starts in the endoplasmic reticulum. Here's a pretty good image of the endoplasmic reticulum. The endoplasmic reticulum is part of the endomembrane system along with the Golgi body, and so it is made of layers and layers of phospholipid bilayers. So it's like little folded up membranes inside the cell. Now, the endoplasmic reticulum is actually an outgrowth of the nucleus. So when we have, in this diagram, an image, we have the nuclear membrane here, and then we have the the endoplasmic reticulum sort of growing out of the nuclear membrane. Now, that endoplasmic reticulum has two regions, the rough ER, which is covered in ribosomes, and the smooth ER, which lacks ribosomes. So for our exocytosis story, we're going to start in the rough endoplasmic reticulum. Now, ribosomes, their responsibility is to read RNA and make protein. So the rough endoplasmic reticulum is one of the major sites of protein production in our cells. And once the proteins have been made, within the endoplasmic reticulum, they can be further modified so that they can be tweaked so they can have the greatest utility for the cell. Sometimes proteins are not quite usable as soon as they're ready. You have to do a few chemical reactions in order to make them usable for the cell. So that goes on in the endoplasmic reticulum. And the proteins themselves could be the cell product, or the proteins could be enzymes which are used to build the cell product. Either way, we need to get those proteins where they need to go, or we need to get that cell product where we need to go, and so it gets shuttled over to the smooth endoplasmic reticulum. Now, the smooth endoplasmic reticulum is responsible for making all of the phospholipids in the cell, and there are lots of organelles that use the phospholipids. The cell membrane needs them, the Golgi body needs them, the endoplasmic reticulum makes them for themselves, the nucleus needs them, and so there are lots of places that need phospholipids, and those all come from the smooth ER. Well, the smooth ER can create transport vesicles. Transport vesicles are little vesicles in the cell that contain maybe the enzymes or the cell product, but they cannot fuse with the cell membrane and have their contents leave the cell. That cannot happen. So there needs to be further modifications. So these vesicles created by the smooth ER end up traveling to the Golgi body. So here in this model we have the nucleus, the rough ER, the smooth ER, and now we have a transport vesicle that is going to the Golgi body. The Golgi body can further modify the proteins or further modify the vesicles, and there's kind of two primary vesicle situations that the Golgi body has. One, it can create a lysosome. If the enzymes in the vesicle that were sent from the ER to the Golgi have a lot of digestive enzymes, if they're digestive enzymes, the Golgi body can say, hey, we're going to modify this, we're going to make this a lysosome. If it has other cellular products or enzymes that need to go to a specific location so that they can do their job or be used, then the Golgi body can create a secretory vesicle. This is a vesicle that can fuse with the cell membrane and release its contents to the cell. So that's what the Golgi body does. So let's review. Exocytosis is exporting large quantities of particles from the cell and is a type of bulk transport. Primarily, we're getting rid of waste and cell products can be a lot of things, such as hormones, sweat, mucus, hair, oils, milk, rubber, pigments, nails, venom, silk, shells, bark, and lots of other stuff. The sweet stuff that living things make. Now, this process starts in the endoplasmic reticulum, which surrounds the nucleus and has two regions, the rough and the smooth region. The rough ER is covered in ribosomes, which receive RNA from the nucleus to make protein. These proteins can be further modified to be useful for the cell, and the proteins might be the cell product themselves, or enzymes which will later create the cell product. These are then moved to the smooth ER. The smooth ER lacks ribosomes, and it's responsible for creating all the phospholipids for all the membranes in the cell. And it will take those materials and prepare a transport vesicle containing them to move those materials about the cell. The transport vesicles, however, are not able to fuse with the cell membrane, but they can go to the Golgi body. And at the Golgi body, the vesicles will be modified. If they contain digestive enzymes, they can be turned into a lysosome. If they contain other cell products, the vesicles can be modified to become a secretory vesicle, which can fuse with the cell membrane and release their contents from the cell. I hope this helps. Struggle well.